Hello, ladies and gentlemen. Welcome to Lecture 2. We're going to talk about the science of psychology. In Lecture 1, we introduced the idea of empiricism in generating data and facts in order to inform your values. This lecture is going to focus on how we actually generate facts. The first part, we're going to review the scientific method. This is the methodology used to systematically address any problem that you want to study. In other words, how do we gather data? The second part, we're going to talk about how we analyze that data using either descriptive statistics, inferential statistics, correlations, things of that nature. And then finally, we're going to talk about what it means for research to be valid and what are the threats to validity. So let's start off by talking about the scientific method. Psychologists, like any scientist out there, are going to use the scientific method to study the problems that they are interested in, whatever that may be. In this case, it's going to be something to do with human thought and behavior. By using the scientific method, we can empirically test our ideas and gather facts to inform our opinions. So just like there are those two major types of psychologists, your research psychologist and your psychologist practitioner, there are two fundamentally different types of research. You have your basic research, which asks very basic questions. How does life work? How do we distinguish different scents, for example? How do we process faces? How do neurons communicate with one another? These are basic ideas. They're not directly related to any major problem. In applied research, this is where we try to provide solutions for known problems. How can we treat depression? How can we treat Alzheimer's disease? How can we better learn information? All right, so here we're applying research to solve a problem directly. In basic research, we're just kind of gathering facts about how life works. Now, these are not two totally separate branches. Oftentimes, labs will have some basic and some applied projects going on simultaneously because the basic research is often going to be used to inform what goes on in applied research. Right? It might come up with new drug targets for the therapeutic companies to go after. As an example here, let's look at some basic research. We're looking at fMRI data, so the functional magnetic resonance imaging. We're looking at brain activity in two different groups of women. On the top here, we're looking at brain activity in women with a low risk of depression. They have no family history. And on the bottom, these are women who have a family history of depression. And what they're doing is presenting kind of sad images for the women to process. Now the colors here, those yellow and red blobs that appear, that's showing you the regions that are activated by sad images. And what should jump out to you here is there's this very distinct area of activity in the high risk group. That area is the amygdala. We're going to become a lot more familiar with this in Lecture 3, but this is the region of your brain that processes negative emotions. And so by having greater amygdala activity in response to these sad images, this creates a much stronger negative emotional response in the high risk group, which maybe explains their depression. So this is some basic data, and maybe we can use this then as we're refining our approach to treating depression. All right, there's our general distinctions. No matter what type of research you're carrying out, you're going to follow the same basic steps. You're going to follow the scientific method here. So the first thing you have to do is identify some kind of a problem. What is it that you want to study? And it could be anything. Honestly, you could apply the scientific method to create the best chocolate chip cookies. But in this case, we might pick something a little more related to science. So let's say it's Alzheimer's disease. That's the problem I want to study. If you're going to study a problem, you need to know something about it. So an important step that's often left out of the scientific method is actually reviewing the literature. What do we know about this problem and what don't we know? For example, you might read several articles and find out that Alzheimer's patients are going to have a buildup of what we call plaques and tangles. These are two different classes of proteins that build up into these large masses. So you see these big balls of protein in the brains of Alzheimer's patients. What you also see is significant neuron loss, or the death of brain cells, and the cognitive impairments or the dementia that the Alzheimer's patient experiences absolutely correlates to the amount of brain tissue that's lost. So you'll collect a bunch of facts and then you'll come up with your hypothesis. What do you think is going on? What do you actually want to test? So based on what we've read here, we might come up with a hypothesis that the plaques and tangles that we see in the brains of Alzheimer's patients are responsible for the neuron death. All right, this is a good hypothesis here, and then we would test it with some experiments. We generate data, and then we would share our results by publishing. So steps 4, 5, and 6 we're going to get to in a bit. Let's have a look at that hypothesis, though. We need to know what constitutes a good hypothesis. All right, it needs to be incredibly focused because it has to be both testable and falsifiable. Now, what does that mean? If it's testable, that means we can actually study it. We can carry out an experiment. And if it's falsifiable, that means that we can prove it right or wrong. All right, this is very important. This hypothesis is definitely testable and falsifiable. We can test whether or not plaques and tangles cause neuron death. So if we put plaques and tangles into a brain and we see neuron death, maybe they cause it. If we don't see neuron death, we know that that's absolutely not the case. We can prove this wrong. The hypothesis that, let's say, the Earth was created in 7 days by an all-powerful being. We can't test that. We can't falsify. We can't prove it wrong. It's not a hypothesis, right? So that's a key distinction for a hypothesis. It needs to be very specific. It needs to be testable and falsifiable. And so that specificity is going to make the hypothesis usually address the relationship between two variables. Sometimes it's more than that, but usually it's somewhere between just two. Now we have two types of variables that we're going to study here. We have the independent and dependent variables. All right. The reason we call these things variables is because they change. When you look at one hypothesis, in this case, plaques and tangles cause neuron death. All right. We can see our variables here are going to be plaques, tangles, neuron death. But not every hypothesis addresses this. Right. You might hypothesize that adding a greater ratio of brown sugar to white sugar will make your chocolate chip cookies softer. That's a hypothesis. You can test it, you can falsify it, you can prove it wrong, right? But notice, we're not looking at the same things. And that's why they're called variables. These things can change. All right, hypotheses always address different things depending on what we're studying. But they do contain these two types of variables. There's our independent variable, and that's what we're going to directly manipulate in our experiment. So in our cookie example, it'd be brown sugar, white sugar. How much are we adding? And the dependent variable is that other variable in our hypothesis that we're actually going to measure. The thing that we're going to see whether or not it's affected by our independent variable. So it might be the softness of the cookie. Right? We change the amount of sugar and we see how soft is the cookie. So that would be our dependent variable. Now if we look at our hypothesis here, our dependent variable is going to be the thing that's going to be influenced by the other. So is it the case that plaques and tangles are causing neuron death in our hypothesis, or are we hypothesizing that neuron death causes plaques and tangles? Well, of course, we're saying that plaques and tangles cause neuron death, so our dependent variable should be something to do with neurons. Like we would count the number of neurons in the brain and see, do they change? What we will directly manipulate will be the presence of plaques and tangles. So plaques and tangles become our independent variable. Right? We can compare brains that have plaques and tangles with those that don't and see if the neuron density changes. So this is a good hypothesis. It's testable, it's falsifiable, and it's very much to the point. As a quick aside, I do want to make it clear that a hypothesis is not a theory, all right, and that your everyday life theory is not the same thing as a scientific theory. All right, hypotheses are very narrow, and they're made to be broken. Right? They are falsifiable. We're there to prove our hypothesis wrong. A theory, on the other hand, at least a scientific theory, is a well-substantiated idea that's based on a large number of studies, and it explains a variety of natural phenomena. All right. It's most likely true, it may or may not be true, but there's a large volume of data that supports this theory. We failed to disprove it so many times that we accept it as generally true. When scientific theories are being generated, we tend to follow the principle of parsimony, where they should be as simple as possible. All right, don't make them overly complicated. Just keep it simple. An example would be the theory of evolution. That all of the species that we see today were derived from common ancestors, and given that they had different mutations and lived in different environments, different traits were then favored to allow them to survive and reproduce. Okay. Another theory would be the theory of heliocentrism, that our planet is orbiting around the sun. Right? This is generally considered true. A scientific law, on the other hand, is regarded as absolutely true in all the conditions that we're talking about. Okay, the law of gravity. Two objects are going to have this attractive force, this attractive pull on one another, depending on how close they are and how massive they are. There's Ohm's law on the bottom there. V equals IR, very important to any neuroscientist or electrical physicist. So an electrical potential, V, is proportional to the current, I, times the resistance, R. As you increase resistance and hold the current constant, you increase electrical potentials. This is a law. It's always true. You can't violate Ohm's law. So if we compare hypothesis, theory, and law, hypotheses are based on sometimes just your intuition. Hopefully there's some evidence. You don't want to waste the time of going through carrying out a study if your hypothesis is total garbage. And so that's why we have that review the literature step. All right, a hypothesis should be based on some evidence. It might not be a large volume, it might be one or two studies. All right. A theory, on the other hand, is going to be based on a large number of studies, and a law is based on even more evidence. All right, we can't disprove it. We know it's true. The focus of these three different concepts also differ. A hypothesis is very focused. All right, there's a very specific topic that it addresses. Neuron death because of plaques and tangles, for example. Scientific theories tend to be more general. Right? The theory of evolution deals with how all of the species alive today have come about. Laws apply to all the cases that they describe. So anytime you have an electrical current and resistance, you're going to have an electrical potential. And so because of what these are based on and what they address, we tend to view a hypothesis as dubious at best. Remember, they are falsifiable. They are there to be disproven. A theory, most likely true, and a scientific law, always true in the cases that are spelled out by it. Right? There's always gravity. You can't escape it. All right, but we don't focus on theories and laws and studies. We focus on hypotheses. We have to test our hypothesis, and we do that using a controlled experiment. Now, if we're going to have a controlled experiment, we need these things called controls. All right, a control is simply a condition where you know what's going to happen. All right, typically what you'll do is either not manipulate the independent variable, or you'll manipulate it in a manner that you know is going to produce the effect you're trying to measure. For example, let's say you wanted to grow more corn, and there's a new fertilizer that came out. Well, you could buy that fertilizer and put it on all your crops and see how much corn you get. That would not be a controlled experiment, though. You have no controls. If you're going to properly test whether or not this new fertilizer is of any use, you're going to need to, first of all, have some corn that you don't put any fertilizer on. That'd be your negative control. No manipulation to the independent variable. We're not putting on any fertilizer. You'll also want a positive control. If you've used a fertilizer in the past, well, go ahead and put that on. Let's see how this new fertilizer compares to your old fertilizer. And so then in the third group of corn, that's the plant that's going to get the new fertilizer. So now we have three groups. We got our negative control, no fertilizer, positive control, the fertilizer you've been using, and the experimental group, the new fertilizer. This allows us to compare the effect of this new fertilizer, right? If we put it on all of our crops, we have nothing to compare against. But hopefully, if it really is better, that new fertilizer grows us a whole lot more corn than our old fertilizer, and hopefully both of those fertilizers grow more corn than no fertilizer. Right? If we don't have those controls, we can't make this conclusion. If they all grow the same, we should conclude that fertilizers aren't doing anything for us. We have plenty of nutrients in the soil, and so we don't need fertilizers. Right? That's a controlled experiment. We have groups that we can compare against. So let's go back to our Alzheimer's experiment here. We hypothesized that plaques and tangles cause neuron death, so we want to look at neuron density in brains that have plaques and tangles and those that don't. So our experimental group is going to be some brains with plaques and tangles in it. That makes the most sense to me. Now, people tend to not like it when you give them debilitating diseases. So you're probably not going to have people for this study. You're probably going to want to use mice. So we'll make some mice and put some plaques and tangles in their brain. Not difficult to do. But if we just look at mice with plaques and tangles, we have nothing to compare against. We need a negative control. Well, that's really simple to come by. Just take any old mouse. Mice don't have plaques and tangles in their brain. So take a regular mouse of the same age without plaques and tangles, compare their neuron densities. But what if we see a difference and it's really small? How do we know if it matters? Or what if we don't see a difference? How do we know that we're able to actually count neurons accurately? It's really easy to write that you'll count neuron density, but it's pretty tricky when we talk about counting thousands or millions of cells. So you want to have a positive control to make sure that if there is neuron loss, you can see it. Simplest thing to do, have another group of mice and inject them with a neurotoxin, any substance that kills neurons. You know you're going to see neuron loss here, so you better have a lower neuron density in your positive control than your negative control. If you don't, you know that your entire experiment has gone wrong and you can't trust any of the data. If you don't have these three groups in your experiment, it becomes difficult to draw conclusions. So if you're going to have a controlled experiment, you need those controls. If you're going to actually carry out an experiment, you also have to have approval. And that approval comes from the Institutional Review Board, the IRB. This is a group of people at the university or company that you work at that reviews every study before it takes place. And their job is to make sure we're not going to violate any human rights. If it deals with animals, there's usually an animal care and use committee that's going to review your procedures to make sure you're not going to cause any unnecessary harm, either to humans or non-human animals. Right? We want to treat all of our animals good, human or not. Now, this wasn't always the case. We didn't always have institutional review boards for psychological studies. These came about in part because of this experiment here, the Stanford Prison Experiment. It's a classic. Let's have a look at it.
At the beginning of this study, the prisoners were arrested at their homes by real police officers and were taken off to the local police station. Now, this was not something which they had signed up for, and they were surprised, understandably, by this event, and they asked the police officers what was the relationship between this event and their signing up for a psychology study. And the police officers refused to be drawn into this conversation. So this was a very unsettling way of starting the study. Zimbardo brought them to a mock-up of a prison, which he created in the basement of the psychology department at Stanford University. What he did then was to divide his group into prisoners, into guards. The guards had a guard's uniform, and they had sunglasses, and they had a stick, and the prisoners wore loose-fitting smocks and sandals. Now, the prisoners spent 24 hours a day in this mock prison. And the guards came and went doing shifts.
Part of what he was doing was observing what was going to take place in this particular context and how those roles as either prisoner or guard, how they shaped or sort of changed the way people behaved.
The guards now fell into their job. Some of them liked it, some of them disliked it, but it was a job that they did. They gave commands, and now what happened is the prisoners followed them.
You can give me the blankets and sleep on the bed and mattress, or you can keep your blankets and 416 will stay in another day.
They found ways to punish prisoners if they misbehaved.
Let's see if we can count backwards here.
Let's try that out.
9, 8, 7, 6, 5, 4, 3, 2, 1. Oh, that's bad. Hey, I don't want anybody laughing.
It was certainly the case that initially the prisoners did show some signs of disbelief and rebellion about what was happening.
This is unbelievable. They took our clothes.
Hands off the door.
But after a fairly short while, they fell into their roles. They became very submissive and dependent, very powerless, and obviously showed signs of stress and anxiety and depression.
Things had gotten so bad that we no longer had a group of prisoners. What we had were individuals believing they were prisoners, struggling for individual survival, no longer caring about their other prisoners or their other fellow students.
Meanwhile, the guards got into their roles, and the level of violence escalated over a period of days. They began to humiliate and degrade the prisoners and find every opportunity to exert their power and influence over the prisoners.
Some of them had prisoners clean out toilet bowls with their bare hands. They now taunt, humiliate, degrade the prisoners in front of each other, and they exert arbitrary control over the prisoners.
This escalation of aggression was so extreme that after a period of 6 days, the experiment had to be brought to a halt before the scheduled time.
I think he showed something that was important at the time, and I think it is about that notion that, because it's not about who you are internally that matters, it's about the social context in which you find yourself.
And there we have it, the Stanford Prison Experiment. It's a classic. All right, let's take a moment and see if we've gotten anything out of this. If we have any understanding of the scientific method, take a moment and pause this recording. We'll come back and answer these in just a second. All right, let's start at the top. So the general steps of the scientific method. First of all, you have to identify your problem. If you don't know what you're going to study, you're going to have a difficult time studying it. I think it's really important to have your next step be review the literature. Find out more about the problem, figure out what we know and what we don't know, and use those facts then to generate your hypothesis that you want to test in your study. After you have a hypothesis, you should be able to design your experiments, carry them out, gather your data, analyze it, and then communicate the results. Oftentimes you'll have to do many studies before you actually publish your results, but that's the basic outline. Find your problem, learn more about it, come up with a hypothesis, test it, communicate your findings. Now, a hypothesis is definitely not the same thing as a theory. A hypothesis is made to be broken. These are testable, these are falsifiable. We're trying to test whether or not our idea is correct with a hypothesis. A theory, on the other hand, is based off of a large amount of evidence, and so theories are made to explain natural phenomena, not to be tested, unlike a hypothesis. So a hypothesis is really what we're using in experiments, and theories are what we create after many, many experiments have been done in a field. All right, so we got a couple different types of variables, and variables are just things that change. Remember, every hypothesis is different. That's why we have variables in them. They're not constants. The dependent variable is the thing that we're going to measure, the thing that depends on our independent variable, and that's the thing that we change. So your hypothesis should include both your dependent and independent variables. You're going to directly manipulate the independent variable and then measure the dependent variable to see if it changes. in order to have a good controlled experiment, you need both controls and experimental groups. All right, so these are both in controlled experiments. Controls are the groups where you have an expectation. You know what's going to happen, right? Because you manipulate the independent variable in known ways. Either you do nothing to it, that's your negative control, or you do something where you know what's going to happen. You use a fertilizer that you're familiar with. You treat with a neurotoxin, right? You know a fertilizer should improve plant growth. You know a neurotoxin should kill nerve cells. So controls, we got negative and positive controls. The experimental group, that's the question. All right, that's the group that you're trying to address with your hypothesis here. So do plaques and tangles kill neurons? Well, I don't know. That's what our experimental group should show us. Is that new fertilizer really that much better? I don't know. That's what the experimental group's going to show us. But the experimental groups mean nothing until we compare them to controls. All right, and those negative and positive controls we've already gone through. Negative controls, you do nothing. Positive controls, you do something that'll give you a known outcome. All right, so both of these are used to compare against our experimental group. And then finally we have those human rights and even non-human rights for animals. Can scientists just do whatever they want? Of course not, right? We have the IRB, we have the Animal Care and Use Committee. So we have groups of people who review experiments before they take place to make sure that all the procedures being carried out make sense. All right, so after we come up with our experimental design, we have to actually carry out the experiment. Right? It's one thing to design one, but you have to actually do it in science. Now, experiments could be purely observational, or they could be real experiments where you directly manipulate something. All right, so these are the two general distinctions. We'll start with observational. Sometimes that's all it takes. You might have an interesting patient come in to see you, and you don't need to manipulate anything. You just simply observe how they function and report your observations. All right, this would be a case study where you look at one or a few people with some rare disease or damage, and you describe how their function is changed as a result of it. So here's an example here. We have a patient SM. It's a fairly well-known patient who has bilateral amygdala damage. We should remember the amygdala. That's that brain structure that creates negative emotions, sadness, fear in this case. Patient SM has damage to both sides of her brain at the amygdala region. She has a rare genetic disorder, and as a result, both of her amygdalas are damaged. As a result, she doesn't experience as much negative emotionality as healthy subjects. Now, if we just look at the top here, we can see that circled region in the comparison, that's the control group here, full of brain tissue still. But over in patient SM, you see in those two red dotted circles, there are two gaping holes, two black regions in the amygdala. That is an absence of brain tissue. So she's essentially missing her amygdala. If we look at her fear response, the kind of light gray bars on the bottom there are showing you the subjective fear rating in response to a number of different clips, different movie clips that should be scary to most people. And you see the light gray bars you see there, they're usually fairly high. It's a scale of 0 to 10. And clip A, for example, elicits a subjective fear rating of 7. So that means that they're a 7 out of 10 afraid. Film clip C, about a 6 or so out of 10. Patient SM would be the dark gray bars. You can see that her fear ratings are consistently much, much lower than healthy controls. In fact, most cases are zeros, with the exception of A, D, and E. She experienced a fear of 1. All All right, so when you have interesting patients like this, you don't need to do any manipulations. You just simply observe and report. A much more common form of observational research would be surveys. Right? You send out surveys to a big number of people to create a decent sample size of the population. So the population is just the collection of all people that you're talking about. So if we're talking about college students, that's our population. If we send out a survey, we're not going to survey every single college student out there. We're going to survey a small sample of them, just a few. Right? Now, depending on the study, you might have a larger or smaller sample size. That's always given as a little n there. So n is sample size. The bigger your sample size, the more likely you are to represent the population. Of course, if we take it all the way up and we do survey every single college student out there, well, now our sample size is the same as the population. All right, and so we know everything about every college student. As we sample fewer college students, we're less likely to represent the population. All right, so you can just observe rare cases, or you can send out surveys. Notice we're not manipulating anything. We're just sending out a survey. Here, answer these questions and send me your feedback. We're not manipulating any real variables here. We're just gathering data. And that data can be quantitative or it could be qualitative. All right, if it's quantitative data, that means it's described using real proportional numbers. All right, that actually means something. You know, if your height is 60 inches and somebody else's height is 80 inches, then they're 20 inches taller than you. All right, if you're only 40 inches tall and another person's 80 inches tall, they're 40 inches taller. They're twice as tall as you. Right? These have a real relationship with one another. Qualitative data can sometimes be described with numbers, but they don't really mean anything other than an order, like a fear rating of 9 and a fear rating of 8. They have a one subjective unit difference, but what does that mean? It can mean something totally different for different people. And is a fear rating of 8 twice as fearful as a fear rating of 4? I don't know. Right? These aren't real numbers. Right? They're 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, but they're just in order. They're really nothing more than adjectives, pretty much. So other bits of qualitative data might be the color of something or how it tastes. When possible, you want to use quantitative data because you can do a whole lot more with quantitative data. It's not always possible, but for example, in those sphere studies that we just looked at, here's some pictures that patients were shown as well. And in fact, patient SM was taken to a pet shop and she handled the snake without any fear whatsoever. You know, she would hold it up close to her face and look at it and say, oh, I wonder what it's thinking. Right? Absolutely no fear. But how do you quantify fear? You know, because it's a feeling. It's very much personal and subjective. So it's a quality about us. A fear of 8 might not be twice as fearful as a fear of 4. It might be 10 times. Who knows? And it varies from person to person. So we want to use quantitative data when we can, because we can do a whole lot more with quantitative data. We can actually analyze it. First, we'll talk about our descriptive statistics. All right, and they are exactly what they sound like. They are measurements used to describe your data. That's it. So if we want to describe the most common value or the most representative value in a set, we have 3 options. We could do the mean. That's where you take the sum, divide by the number of items. It's just your good old average. We could state the median value, right? If we put all of our values in order from low to high, we take the middle value. That's our median. Or we could say the most common value. That'd be the mode. Which value do we see the most? Now, under ideal conditions in a normal curve, that'd be part B here in this illustration, the bell curve, the normal curve, the mean, median, and mode are all the same. Right there at the peak of that bell curve, that's the mean. That's the 50th percentile, so that's the median, and that's also the most common value, so it's the mode. In reality, you don't find normal distributions. You find things that are close to it, but they're never going to be perfect normal curves. Instead, you're going to have a little bit of skewing. Heck, man, sometimes you see more than one peak in your data. But we can see in parts A and C, sometimes you have negative or positive skewing, and when that happens, your mean, median, and mode are not the same value. The most commonly used value would be the mean, so we're going to focus on that. And besides describing the most common value, we also need to indicate how variable the data are. So you could simply report the range of values. What's the difference between the lowest and the highest? The interquartile range would be the difference between the 25th and 70th percentiles, but the most common value for variability would be standard deviation. Now, before your eyes just glaze over at this equation, it's really very simple. It's the sum of squares for the difference between every value in the mean divided by the n, and you take the square root. All right, you don't need to know that, but what it essentially is the average sort of difference between all the values and the mean. So in this case, the little sigma there is standard deviation, and that mu would be the mean of your sample. So in a normal distribution, which is what we see in this image here, the mean is right there in the middle. And as we move 1, 2, or 3 standard deviations away, you can see we're moving further away from the mean, and we encompass different amounts of the bell curve. Now, whenever we look at standard deviations, this is telling us essentially how wide is our bell. Right here, if we compare different standard deviations, you can see that as the standard deviation increases from 5 to 10 to 20, that bell curve, even though it's still a normal curve with the same mean, flattens out. It gets a little shorter and a little wider. Right? It becomes a whole lot more variable. We're going to be calculating means and standard deviations in our basic statistics Google sheet. So you'll become a lot more familiar with these later on when you go through that exercise. So let's say that we gather a bunch of data. What can we do with this? Well, sure, we can describe the mean and the standard deviation, but we can also try to describe relationships between two different variables by looking for what we call correlations. So in this hypothetical correlation here that I've plotted, we have our independent variable on the x-axis, that's where they always are, and our dependent variable on the y-axis, again, that's where they always are. And you can see as the independent variable increases from 1 to 2 to 3 to 4 to 5, so does the dependent variable. And if we draw a straight line through it, you'll see it hits every point. And right at the top, we get our R value. That's the correlation coefficient, R. It ranges from -1 all the way up to +1, and so this one happens to have a correlation coefficient of 1. That means it's an absolutely perfect positive correlation. As the independent variable increases, so does the dependent variable. An R value of -1 looks like this. This is a perfect negative correlation. As the independent variable increases, the dependent variable decreases. Right? They're not moving in the same direction. As one goes up, the other goes down. Now, it's not always the case that you have perfect correlations. Right? It's going to be a range anywhere from -1 to +1. A correlation coefficient of 0 means that there's absolutely no correlation. But typically you find some value reported. Now anything greater than 0.8, or if it's negative, less than 0.8, is a strong correlation. So here we have a strong positive correlation. As the independent variable increases, so does the dependent variable. It's not perfect, but you'll see overall there's this positive trend here. This would be a strong correlation. A weak correlation is going to be anything less than 0.5, right? Or greater than -0.5. It's a negative correlation. For example, this R value of -0.11. That's a very weak negative correlation. If you look, it really doesn't look like there's any relationship here at all. Of course, when we do the analysis, you get a number, But because the R value is so small, we take this to mean that if there is any correlation, it's very weak. Now that correlation coefficient doesn't just tell you how good the correlation is. We can also use it to estimate how much one variable influences the other. And all we have to do there is square the value. So if we square the R value, we create our coefficient of determination. In other words, what proportion of the variance in our dependent variable is caused by the change in our independent variable. So if we change the independent variable, how much change should we expect in the dependent variable? So if you have a nice, strong, positive correlation of 0.922, if you square that, you'll get 0.85. What this means is that 85% of all the variability that you see in the dependent variable is explained by the independent variable. All right, so only about 15% of the change is due to something else. In other words, you got a pretty good explanation going on here. In this weak negative correlation, well, the correlation looks like crap, and if we square negative 0.11, we get roughly 0.01. So that means that at best, only 1% of the change in our dependent variable is caused by the independent variable. In other words, this is meaningless. Move on. All right, nothing to see here. We have no correlation. Now, just because you see a correlation, that does not in any way prove causation. Now, here's what I mean by that. Let's look at our hypothetical data in the upper right hand. What we see here is if we look at the x-axis, that's the amount of time spent watching violent TV. The y-axis is some arbitrary rating of aggression, where higher values mean more aggressive behavior. And you can see there's somewhat of a positive correlation here between the amount of time spent watching violent TV and the degree of aggression. And so when you look at this, you might conclude, hey man, if you watch a lot of violent TV, that's going to cause you to be aggressive. Well, that might not actually be the case. It's just as plausible to say, well, maybe it's the case that aggressive people tend to watch more violent TV. That's plausible. That's consistent with our data. It could also be the case that it's more complicated than that. If you have a little bit of aggressive behaviors, maybe you want to watch some violent TV, and then by doing so, well, that increases your aggression. Well, then that makes you want to watch even more violent TV, and you become even more aggressive. So maybe they're actually acting on each other. It doesn't have to just move one way. And of course, it could definitely be the case that these don't actually directly influence one another. Violent TV watching and aggressive behaviors might not be directly related. There might be something else that accounts for that. Maybe it's just bad parenting. Bad parents are making them view violent TV because they don't care. They just want to be left alone, and maybe the bad parents spank the kids too often, and that makes them aggressive. So there might be something else, some other variable that we haven't studied that accounts for this relationship that we see. And those unstudied variables that we don't know about, that's what we call confounding variables. These are very important and absolutely need to be addressed in any good study. Right? Confounding variables are the basis of alternative hypotheses. So when we see our data up there, the more time spent watching violent TV is related to an increase in aggression. Okay. We can't argue with the data. We can argue about their relationship. Does one cause the other? Do they act on each other? Or is there a confounding variable? Now, of course, there's the final option that this is all bullshit. These two variables actually have absolutely nothing to do with each other, and it's just by chance that you see this correlation between them. That's what we call a spurious correlation. Let's see a few examples. Here we're looking at two different pieces of data. In black, we're looking at the number of suicides by hanging, strangulation, and suffocation. And in red, we're looking at the amount of money the U.S. spent on science, space, and technology. And we're looking from 1999 to 2009. As you can see, these two lines move pretty well together. Does this mean that as we spend more on science that people are killing themselves by hanging? No. Does it mean that as people kill themselves more by hanging that the U.S. decides to spend more on science? No. All right, there's absolutely no relationship between these two things. This is a spurious correlation. Another example, the number of films that Nicolas Cage appeared in. At each of these years is shown in black, and you can see in some cases this lines up pretty well with the number of drownings that occurred from falling into a pool. You know, in 2007 there, we see a spike in Nicolas Cage movies. And there's also a spike in drownings from falling into a swimming pool. And then when Nicolas Cage takes, a slower year there in 2008, okay, drownings return back to normal. Does this mean that people are drowning themselves as a result of Nicolas Cage movies? I highly doubt it. This would be a spurious correlation. And one final one, drownings, this time from falling out of fishing boats. You can see that correlates somewhat with the marriage rate in Kentucky. Are these related? Absolutely not. They're just spurious correlations. So correlative experiments and observational experiments are not as powerful because you're not directly manipulating anything. You're just looking at the data and trying to describe mathematical relationships which may or may not be real. It's much more powerful for you to take the experimental approach and directly manipulate your variables. All right, in this case, the independent variable. So in any good experiment, what you're going to do is systematically change one thing, all right, and then measure its effect. So in this case, we're gonna make a single manipulation to the independent variable and see how that affects our dependent variable. And by changing just one thing, this helps us control for confounding variables. Now, in an ideal world, we'd like to test the same group of subjects multiple times, right? This is what we call within subjects design. By using the same subjects multiple times and systematically changing the independent variable, we know that we've controlled for most, if not all, confounding variables because we're using the same people. This isn't always possible. Sometimes you have to use different groups. All right, that would be a between subjects. So in this case, you'll compare how two different groups measure whenever they have two different versions of the independent variable. All right, we'll see an example of this in just a moment. Now, if you're going to make different groups, typically this is done by random assignment. With a large enough sample size, the assumption here is that by randomly assigning different people to different groups, we can pretty well control for confounding variables like differences in age, education, personality, things like that. Sometimes more complex assignment methods are used, but random assignment is the most common. So let's see the difference between within and between subjects. All right, that might help make things clear. So let's say we wanted to see whether or not a new smart drug had any effect on people's IQ. All right, so in within and between subjects, well, we have to have a group where we just measure the IQ, no smart drug. All right, now if it's a within subjects design, we're going to take those same people, in this case the blue and the green people there, and now we'll measure their IQ after we give those same people the smart drug. So any differences in their learning ability or their personality, any of those potentially confounding variables, they should be irrelevant because they're the same people. There should be no confounding variables between these two IQ measurements. The only thing that's changed is that they've been given a smart drug. Well, that's not really the only thing that's changed. In fact, remember that testing effect? Since we're measuring their IQ a second time, maybe they're just getting better at IQ tests. So in order to control for the testing effect, we might use a between subjects design. So in this case, we'll measure the IQ in the blue and the green people, no smart drug, and then we'll take a different group of people, hence the different colors here, and we'll give them the smart drug and we'll measure their IQ. Either one of these approaches in isolation isn't going to be perfect. But by doing both of them together, you can have a much more powerful experiment. So the between subjects design can account for the testing effect. The problem is that you have a ton of potential confounding variables, differences in the intellectual function of your two different groups. If you don't have some baseline measure, well, then that won't work. So what you'll probably do is have a combination of these, where you'll have your initial baseline group with no smart drug made up of your blue, green, orange, and red. Then you take the blue and the green and you measure their IQ again without the smart drug, and the orange and the red, and measure them with the smart drug. So you have a combination of the two. It's not always one or the other. But if you have to choose, take the within subjects whenever possible. That's going to do the best job of controlling for confounding variables, just innate differences between people or animals. So after you've gone through the trouble of carrying out your experiment and generating all of your data, you got your experimental groups and your control groups. You want to see, is there any difference? Well, now we have to turn to inferential statistics. We're not going to go into these in any level of detail. And again, before your eyes glaze over here, the t-test is the most common statistic. The formula is given here. In this case, that big X with the line above it, that's your mean for group 1 or group 2. The subscript tells you the difference. S would be the standard deviation, and N is the sample size. So you can see that the t statistic is going to be based on the difference between your two groups. So the bigger the difference, right, the larger the t statistic. And it's also related to your sample size and your standard deviation as well. We're not going to need to use this formula. You will carry out t-tests in the Basic Stats Google Sheet, but what you're going to get at the end is the p-value. That's what we care about. We don't actually care about the t-value per se. We care more about the p-value. P-value is the probability value, and it's the probability that your two groups are not different. All right, so the higher the p-value, the more likely it is that any differences you see are not real. All right, so if you see a slight decrease in neuron density with plaques and tangles, and your p-value is 0.5, that's telling you there's only a 50% chance that this difference is real. What we're trying to look for, the value that you need to get familiar with, is 0.05. All right, 5%. P has to be less than 0.05 for us to believe that the difference is statistically significant. In other words, that we should believe that we have a legitimate difference. So if there's less than a 5% chance that the differences you see are due to chance, we believe that is real. All right, so if we do the same experiment, we compare the neuron density in brains with plaques and tangles to those without. We see that there's a pretty big difference. We do our inferential statistics. We get a p-value of 0.001. That's definitely less than 0.05. That's saying that there's a 1% chance, a 0.1% chance that those differences you see are not real. That means we're 99.9% correct that there is a difference. So we should believe that. That seems pretty good. So the lower the p-value, the better. Anything less than 0.05 we take to be real or statistically significant. Again, you'll get a lot more on this whenever you do the Basic Stats Google Sheet. But that probability value, all right, from the t-test or any test, there's a bunch of tests, not just the t-test. That's the most common, that's why I put it here. But in the end, what you care about is the p-value. All right, let's take a moment and review what we've learned. We'll answer these in just a moment. Go ahead and pause the video. All right, let's see what we can do to distinguish our sample and population. Well, the population is the entire group of people or animals or plants that you care about. So again, if we're going to study the elderly, people 65 and above, that means our population of interest is everyone 65 and above. Now, we won't be able to do an experiment on every single elderly person, so instead we select a small group of them, and that's our sample. We hope that the sample represents the population, but it doesn't always, right? If you take any 20 elderly people out there, they probably don't represent every single elderly person. All right, so that's why we need a large sample size so that we can hope to at least closely describe the population from our study. Then we got that mean, median, mode. Well, these are all used as descriptors for a data set. They're the representative value, if you will. The mean is the arithmetic mean, the sum divided by the number of samples. The median is that 50th percentile. All right, the middle value, and the mode is the most common value. All right, if you just count them up, the mode is the one that's present the most number of times. The standard deviation is a measurement of variability. The larger the standard deviation, the more variable your data are. The standard deviation very basically is a somewhat average value of difference between all the data points and the data set's mean. All right, have a look at the formula. You'll see how it's calculated, but it's just a measure of variability. Confounding variables would be those variables that we didn't look at in our study that may potentially explain the differences that we see. Right, so in the example of the violent TV and aggression, bad parenting would be the confounding variable. The way that we control for them is with careful experimental design. And the best way that you can do that is to try to use a within subjects approach. Now, in correlational research, correlation absolutely does not prove causation, and the reason for that is severalfold. We're trying to say that variable X affects Y. Well, maybe it's the case that variable Y affects X. You'd see the same correlation. Maybe variables X and Y affect each other. Maybe there are confounding variables. And maybe there are spurious correlations. All right, don't forget, pure chance. All right, within and between subjects. You can come up with any examples you want here. Within, you're just going to use the same subjects in multiple cases. So one example might be if you're going to test whether or not a new mattress actually improves back pain. Within subjects design would have the same group of people sleep on their normal mattress that's giving them back pain, and then to have the same people then sleep on the new mattress and have them compare their relative amount of back pain. Between subjects would use two different groups of people. One group of people sleep on their normal mattress, another group of people sleep on the new mattress, and then you compare the amount of back pain between these two different groups of people. The major downfall for the between subjects design is all those potential confounding variables, differences in health between subjects. All right, and finally, that p-value. What does that represent? Well, p is probability. So the p-value represents the probability that the differences between your two groups are due to chance. So the lower the p-value, the more likely it is that your differences are real. The typical cutoff value is 0.05. What that means is that there's only a 5% chance that the differences you're seeing are due to chance. In other words, we're 95 or more % certain that the differences we see are because of a relationship between the two variables that we're testing. So any p-value less than 0.05 is taken to mean statistically significant. The difference is real. All right, finally, we're going to talk about research validity. All right, it's great to carry out an experiment and to use the proper design, but experiments are really only useful if we can believe the data and the conclusions that we draw from them. In other words, the work has to be valid. Well, how do you know if work is valid? Luckily, you don't have to worry about any of this. All right, that's the purpose of peer review. So if you're reading a published manuscript in any peer-reviewed journal, you can feel pretty confident that what you're reading is valid. all right, because there's a couple of steps that you have to go through if you're going to actually publish your work. The first thing that you do after you carry out your experiments and you think you have a story to tell, you send your manuscript to an editor of some academic journal. For example, if you're working in the field of neuroscience, you might send it to the Journal of Neuroscience. We see the cover over there, very pretty picture. If you're a psychologist, you might send it to the Journal of Psychology. And every one of these journals has their own editor who's going to read your cover letter, glance at your manuscript, and decide whether or not it's of potential interest to the journal. All right, they don't judge your work for validity. All right, editors can't know everything in the field of psychology or neuroscience, so instead what they do is send the work out to a collection of experts that we're going to call reviewers here. And these are people who work in the field related to the manuscript, who can review it to look for validity. So if you finish that study on plaques and tangles and neuron death, and you send your work into the Journal of Neuroscience, well, your reviewers are probably going to include people who know about plaques and tangles or Alzheimer's disease or neuron death, so they can look at your work and see, are there any flaws? And if there's nothing wrong with it, congratulations, your work is published, and it'll show up in a coming issue of that journal. It's usually the case, though, that there are at least some flaws and the work is rejected. It might be the case that it needs a little bit of tweaking and then it will be accepted. And it might be the case that it's so much work, it doesn't make sense to continue with this journal. You try a different journal with different reviewers and hope for something better. If the reviewers have very valid points, you might just need to perform more experiments. Maybe they thought of some confounding variables that you didn't, that you need to go test and make sure they don't account for what you're seeing. So when you're a reviewer, one of the things you want to think of is construct validity. Did the variables that they actually measured have anything to do with their conceptual variable of interest? For example, if we're going to measure intelligence, all right, over here, this is a question from an IQ test. Well, is this really intelligence, or is this spatial reasoning? Is spatial reasoning a very big part of intelligence? This is where you have to try to determine, did they measure the right thing? In our Alzheimer's experiment, measuring neuron density, well, that makes good sense. What if we just measured the size of neurons? Does that matter? Probably not. What if we looked at the size of the rat's head or the mouse's head? Does that matter? No, that doesn't tell us anything about how many neurons are there. The weight of the rat wouldn't matter. So in order for our measured variables to be valid, we need reliable measurements, and we have to actually measure the right thing. And this is, again, why controls are really important. If you're doing neuron counts and you see 1,000 neurons in one brain, that's way too few. And if you see 10 trillion neurons in another brain, that's way too many. Yeah, you're seeing very different values here. You should see similar numbers of neurons in all of your control brains, right? Like if you stepped on a scale, you'd want to get the same weight, ideally, every day, right? You wouldn't want it to fluctuate from 10 pounds to 110 pounds, right? So we need to have reliable measurements that actually measure what we care about. And whenever we're reporting our data, in our Alzheimer's study, if we say there are fewer neurons with plaques and tangles, unless we actually measure memory function in that animal, we can't say that these animals have any reduction in memory function. We can just say that there are less neurons. We can't say plaques and tangles cause memory loss. No, we can say instead that plaques and tangles are causing neuron loss. Those are related, but different. The next question you might ask is, did we use the right statistics? All right, so all of these statistical tests that we do have underlying assumptions. Typically, we assume a large sample size, oftentimes 20 or even 40 samples are needed in each group, but you don't always see that. Some studies, you're going to have maybe 3 or 7 for your sample size. We also assume a normal distribution like we see over here on the right. Hopefully we remember this confidence influenced by feedback graphs from the first lecture. We're assuming a normal distribution for these samples. Were they? I have no idea. They didn't report that data. All they gave were means and standard deviation, so I reconstructed these curves. In order for the test to be valid, it needs a normal distribution. That's what a t-test assumes. If they're not, you need to use a different test. We assume similar variability in the groups. We assume a linear relationship between our two variables that we're testing, right? And so you can violate some of these assumptions, but if you're violating all of them, then you probably need to use a different statistic, right? And this is another thing that reviewers will look at. Next, we need to think about whether or not we can trust whether or not we have a causal relationship. Remember, correlation doesn't prove causation. And so what we need to think about, are there potentially some confounding variables that can explain this? In other words, are there alternative hypotheses? So to rule out, potential confounding variables, we do a number of experiments. And we also have to be very careful with our experimental design, especially if these studies involve humans. And every study, at least to some degree, does. There's at least a human carrying out the study. So we use this method called blinding in order to help rule out some potential confounding variables and biases. So in a single blind study, that means that one group is blind. They don't know what's going on. In this case, it's the subjects. And this is a very important point here. The placebo effect is absolutely real, and we absolutely have to control for it. So whenever we do any sort of drug study, we have to include placebo controls, because just simply expecting an effect can cause us to believe that that effect has occurred. All right, so if we're dealing with subjective feelings like pain, we need to be very careful and have the proper placebo controls. So if you think that there's a new drug that helps improve lower back pain rather than that mattress, well, you need to give out a placebo. You need to have a placebo control group. So one group of people that you give a harmless sugar pill. That's your placebo. This should do nothing to back pain. And another group that you give the actual pain-treating drug. Because simply expecting your back to feel better could be enough to make you trick yourself into having less back pain. So placebo controls can help rule out confounding variables. The placebo effect, you'll actually see this in many studies that still get through review. I'm not sure how, but they do. You'll see a lot of drug studies without placebo controls, and when you see that, you should immediately just go ahead and put that study down and take nothing away from it. A much more powerful approach is to use the double-blind study, because not only do the patients or the participants in the study have their own expectations, so do the researchers. Everyone wants to be right. They want their studies to work. They want to be able to publish their results and have their careers move in a positive trajectory. So the researchers really want to be correct. So whether they consciously do this or not, everyone is subject to bias. And so we have to control for the bias of the experimenter. And that's where blinding them comes into place. So now in a double-blind study, it's not just the one group who doesn't know what's going on. Both groups are blinded. So neither the subject nor the researcher who's observing them knows what group they're in. Right? If you know that you've given one group the placebo and this other group the drug that you're trying to publish as being very useful so you can get some money for this new pharmaceutical treatment, well, you might rate how that group is behaving in a manner that you see fit. You might just say, oh yeah, it seems like they have less pain, even though they don't. So in order for your study to be internally valid, what you need to do is to be double-blinded. So the patients don't know which pill they're getting, and the researcher who's recording their observations don't know which pill each patient got. So if you don't know the difference between placebo and drug, and you consistently report that the patients who received drugs had lower pain, then you can put a lot more faith in that observation. Then you know it's not just your own bias leading you to draw this conclusion. So double-blind studies are really the gold standard if you want to achieve internal validity. Then finally, it would be nice if your study on the sample that you've looked at actually relates to humans in general, right? Do you have external validity? Do the results that you report here actually apply to the general population, or does it only apply to the specific sample that you studied? Right, for example, if we can only recruit 18 to 20-year-old American males, can we generalize their results to elderly women or babies or people in different countries, or in this illustration here, Buddhist monks, right? Right, if we got this guy here who's drop kicking who knows what, essentially shirtless with a tie on, does his mind function the same way as this guy over here to the right who's meditating? You know, how similar are they? At a biological level, there are probably some good similarities between them, but once we're talking about at a cognitive level with their thought processes, they're probably very different people. So your external validity can really only be proven by replicating your study in a different population, right? So if you believe that there's a better way of treating depression in Western women, okay, that's great, but does it treat depression in men? How about in people in other cultures? So repeating the experiment helps increase external validity, but it takes a long time. A much faster way of showing external validity is to do what we call a meta-analysis. And that's to look at studies that have already been done. So if somebody has looked at depression treatments in many different populations, or for example, whether or not ginkgo biloba improves memory function in different populations. By looking at all those studies, a meta-analysis takes their results and then essentially puts them all together and says, based on these studies, most find that there's a difference, and so we should believe that this finding is externally valid. So if you see something multiple times in multiple populations, then you should start to believe that this is generalizable. This is not just something specific to the population you care about. All right, let's go ahead and see if we've gotten anything out of this. Take a moment and answer these. We'll pause the video and come back in just a second. And we're back. All right. Peer review. Well, this is where your peers, and by peers I don't mean folks who are the same age as you. I mean people working in the field that you've studied are going to review your work. So remember, you send your manuscript off to a journal and the editor sends it out to those reviewers. In peer review, experts in your field determine whether or not your findings are valid, because there are a few threats to validity that we have to keep in mind. So first of all, there's the construct validity. Did we measure the right thing? Right? Should we believe the numbers that we've gathered? There's the statistical validity. So did we use the right test? Did we analyze our numbers appropriately? There's internal validity. Right? Should we believe these conclusions? Are there potential confounds? So confounding variables or bias either from the participants or the researcher themselves. And finally, there's the threat of lacking external validity, right? Does the phenomenon that you're reporting here only happen in the specific group that you studied, or does it generalize to human beings as a whole? All right, that about does it for Lecture 2. I'll see you guys for Lecture 3, where we talk about nervous system function and the human brain.